What is the structure of baculoviruses? If you're curious about the tiny but powerful world of viruses, particularly those that affect insects, you're in the right place. Let's take a closer look at the structure of baculoviruses, which are quite fascinating. Baculoviruses are a family of viruses that primarily infect insects, especially the larvae of moths and butterflies. These viruses have a unique structure that helps them survive and spread. First, let's talk about the genetic material. Baculoviruses contain a circular, double-stranded DNA genome that ranges in size from 80 to 180 kilobase pairs. This DNA is enclosed within a structure called the nucleocapsid. The nucleocapsid is made up of a major capsid protein called VP39, along with some minor proteins. This protein forms a covalently cross-linked helical tube that protects the DNA. The ends of this tube are sealed by base and cap substructures, which are crucial for the virus's entry into the host cell. When we look at the virus under a microscope, we see two distinct forms, the occlusion-derived virus, ODV, and the budded virus, BV. The ODV is embedded in a protein matrix, either polyhedron or granulin, which forms a highly stable protein crystal. This crystal dissolves in the alkaline environment of the host insect's midgut, releasing the virus particles. The BV, on the other hand, is released from the infected host cells and is responsible for spreading the infection within the host. It acquires its envelope from the host cell membrane and contains viral glycoproteins such as GP64, which are essential for the virus to enter new cells. The GP64 protein is particularly important because it facilitates the budding of the virus from the host cells and its transmission from cell to cell. This protein forms structures called peplomers on one end of the BV particle and is responsible for the pH-mediated envelope fusion to the endosome, allowing the virus to enter the host cell. In terms of the viral envelope, BVs and ODVs have different compositions. BVs have an envelope consisting of phosphatidylserine, while ODVs contain phosphatidylcholine and phosphatidylethanolamine. The life cycle of a baculovirus involves several phases, including early, late, and very late phases. During these phases, the virus replicates within the host cell nucleus and new virus particles are produced. The ODV form is produced in the very late phase and is embedded in the matrix of occlusion body protein, making it highly resistant to environmental factors. Understanding the structure of baculoviruses is crucial for their use in various applications, such as biopesticides and the production of recombinant proteins. These viruses are highly specific to their insect hosts and are safe for use in controlled environments, making them valuable tools in both agricultural and medical research.